I'm going to reacquaint you with the timeline that we already talked about in the first lecture. You're definitely going to need to have watched that first lecture in order to understand this one, but let me just establish this for you. So, in our connection between Jewish individuals and the building out of this concept of both viruses and vaccination, which is pushed us toward where we're at now with this mandatory aggressive push for experimental vaccines in 2020. So we have the first Jewish person was admitted to the Royal Society in 1732. We know Jews were admitted as Freemasons in the mid 1700s, and at that point, Edward Jenner was both a Freemason and a Royal Society member prior to his first implementation of handing out vaccinations in 1796. Then we see another Jewish individual, Jacob Henley, producing this concept of contagion vivum. The first vaccine certificate is issued to a Jewish man, Salomon Pollock. Mordecai Wolf gives his cholera vaccine to India. Then we see inconsistent results with tetanus and diphtheria testing in 1830. Paul Ehrlich, who's Jewish, works with these serums and is considered the father of and generates the basis of modern immunology. Then Maurice Fishberg comes out in the same year and says there's no Jewish opposition to getting vaccines. In other words, at least from an outward appearance, Jewish people are in favor of getting vaccines. Then Karl Landsteiner discovers the polio virus, and I touched on Jonas Salk, but we'll go into more detail with Albert Sabin in this lecture. We looked at Harry Rubin and the RSV viruses that supposedly cause cancer. We looked at Bernal's work with X-ray crystallography, and we finished up with Rosalind Franklin. That's where we left off in the last lecture, and I sort of poked some holes into basically every single aspect of these individuals' works, largely by referencing previous lectures that I've already done. So what we still need to cover is the works of Salvador Luria, who came up with this concept of replication mechanism for viruses, how they replicate. We'll see that he doesn't actually propose a specific replication mechanism. Then another Jewish scientist, Temin, comes up with this concept of reverse transcription. Again, this all provides increased justification for more and more invasive technologies. We see the discoverer of the HIV virus is a Jewish man, Willie Rosenbaum, and experimental and dangerous drugs were prescribed to those individuals as a result. Then we look at the work of Stanley Falco, who generates Koch's molecular postulates, which makes things even more aggressively virtual and computer generated. Then we see the work of Jeffrey Taubenberger, who retroactively goes back and identifies the Spanish flu of 1918 and says it's really H1N1. And then we'll touch on the work of Drew Weissman, who is the individual that published the original papers that were used as the justification for the COVID vaccines, and of course he's Jewish. Then we'll look at the sort of policy dictators that pushed policy on how the COVID vaccines were to be distributed, and et cetera, et cetera. We'll see Israel has an outsized role in that, and that many of these health agencies, of course, were headed by and policy was dictated by Jewish people. So with all that said, let's start out with the discussion of Mr. Salvador Luria here.
So now we move on to Salvador Luria. What was his contribution to the virus idea? Well, he was an Italian microbiologist, later a naturalized U.S. citizen. He won the Nobel Prize in Physiology or Medicine in 1969. With Max Delbruck and Albert Hershey for their discovery on the replication mechanism and the genetic structure of viruses. He showed that bacterial resistance to phages is genetically inherited. So those are some bold claims, right? So he was born as Salvatore Luria in Turin, Italy, to an influential Italian Sephardic Jewish family. His parents were David and Esther Sacerdoti Luria. He attended the medical school at the University of Turin, studying with Giuseppe Levi, which is, of course, a Jewish last name as well. There he met two other future Nobel laureates, Rita Levi-Montalcini and Renato Del Becco. He graduated from the University of Turin in 1935 and never got a master's degree or a PhD, as they were not contemplated by the Italian high educational system, which was very selective. At the University of Rome, he was introduced to Max Delbruck's theories on the gene as a molecule and began to formulate methods of testing genetic theory with bacteriophages viruses that infect bacteria. In 1938, he received a fellowship to study in the United States, where he intended to work with Delbruck. Soon after he received the award, the fascist regime of Mussolini banned Jews from academic research fellowships. So he began this phase research in September 12, 1940. His famous experiment with Delbruck, known as the Luria-Delbruck experiment, demonstrated statistically that inheritance in bacteria must follow Darwinian rather than Lamarckian principles. He published a quantitative analysis of the effect of ultraviolet radiation on bacteriophage multiplication. During the early course of infection, they found an increase in bacteriophage resistance to ultraviolet radiation, and then later a decrease. At the time, this pattern, known as the Luria-Leiter-Jet effect, was published. Little was known about the central role of DNA in biology. Later work established that multiple DNA repair pathways encoded by infecting bacteriophages contribute to the increased UV resistance early in infection. And so they're referencing this paper published in 1993. He worked with James Watson, who went on to discover the structure of DNA with Francis Crick and became a naturalized citizen in the U.S. in 1947. So again, everybody's sort of orbiting Watson and Crick with their DNA ideas in this story. So Luria moved to Illinois-Urbana in the 50s. Luria and Bertani discovered the phenomenon of host-controlled restriction and modification of bacterial virus can significantly reduce the growth production of phage growth in strains. He became chair of microbiology in 1959. He switched his research to focus from phages to cell membranes. He found bacteriorhizins impair the function of cell membranes. 1972 became the chair of Center of Cancer Research at MIT. So it discusses all these awards that he won, and they say he discovered the replication mechanism, but notice there's no specific citations for that. Here's the research works they provide us. They provide us with mutations of bacteria from virus sensitivity to virus resistance is the title, mutations of bacteria and bacteriophage, ultraviolet radiation of bacteriophage, non-heredity host-induced variation of bacterial viruses, host-controlled variation in bacterial viruses, and that's basically it for those papers. So none of them explicitly reference this replication mechanism he supposedly discovered. So here we have ultraviolet irradiation, and what are we doing here? So what did they do? They took E. coli strains, and they put them on Petri dishes, and they heated them for 40 minutes at 37 degrees Celsius. It had a latent period of 13 to 14 minutes before 25 minutes and a yield of 40 to 60. So what are we seeing here? They're irradiating this sample with UV radiation in a Petri dish, and they're watching it produce particles, and they're saying this is somehow indicative of multiplication of this stuff, killing of non-infected bacteria in a free phage. The survival curve in synthetic medium was determined down to a survival of 10 to the minus 4 using clear non-screening suspension. Once again, what are we seeing here? Just like we saw in all the previous experiments. None of this is being done inside of living animals. They're looking at Petri dishes, and then they're making massive assumptions. Let's look at another paper that he did. Host-controlled bacteria variation in bacterial virus. That sounds promising because if you have a host, it needs to be an animal or a human being, right? So they talk about non-hereditary mechanisms have sometimes been postulated. So they took an E. coli strain of a non-lysogenic derivative strain of E. coli. And what are they doing? They're doing cultures again. That means it's inside of a Petri dish. And they isolated it from this lysogenic strain, and they referenced this Bertani process from 1951. Then they added calcium for adsorption between these two samples. So again, they're just synthetically adding stuff inside the Petri dish. They're basically, I would argue, they're torturing the sample until it gives them the results that they want. But you can go ahead and disagree with that. Then they're adding magnesium, and they're, testing this. Again, all of this is withdrawn and separated from a human or animal body. And they're doing multiple passages of this, and they're saying, look, it's changed the structure of the quote-unquote virus, When in reality, I mean, you're just playing with genetic material inside petri dishes and exposing it to different things, and then extrapolating that has any sort of relationship to the human, what's happening inside the human body. Mutations of bacteria from virus sensitivity to virus resistance. When a pure bacterial culture is attacked by a bacterial virus, the culture will clear after a few hours due to destruction of the sensitive cells by the virus. So what is this? This is the same thing as cytopathic effect. So they're saying they took a pure bacteria culture and they're exposing it to this bacterial virus and it's killing it all. Okay, so this is 1943. They grow a bacterial culture on a single cell. The culture is plated with virus in excess. We haven't even talked about how they're getting this virus. One finds that a very small fraction of the bacteria survive the attack of the virus, as indicated by the development of a small number of resistant colonies consisting of bacteria which did not even adsorb the virus. Where are they getting this virus from? They have these different hypotheses, and they suggest all these hypotheses. Here's the theory, blah, 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 that they're suggesting over and over and over and over and over again. And what I'm searching for in this entire paper, finally we get to materials and methods, more than half the way in. Just to highlight, though, for a second, at least they're trying, because this is 21 pages with calculations. When we looked at the DNA discovery, the structure of virus as identified by
Rosalind Franklin,
using X-ray diffraction, it was just, here it is in 2 pages, we've established this, and it's definitely a double helix structure. So notice with Delbruck and Luria, at least they're explicating this over 20-something pages, so we can think that there's at least some sort of scientific process going on. But they took E. coli and secondary cultures after apparently a complete lysis of B by virus A shows up within a few hours. What viruses are we talking about here? So cultures were grown either in a nutrient broth containing 5% NaCl, asparagine glucose synthetic medium, 35 minutes as compared to 19 minutes in broth. Synthetic medium, the acidity increased during the time of incubation from a pH of 7 to a pH of 5. So again, they've removed everything from the realm of what it would actually be like in the human body. So how can you extrapolate this out to what's actually taking place in human beings? And as far as I can tell throughout this paper, I can't tell how they're getting these supposed viruses isolated into the bacteria in the first place. They don't really describe this very well, and they openly admit they're using a synthetic medium, which means this isn't in any way representative of what it's like inside the body of a human being or of an animal. So how can we extrapolate the results this way? And then they're developing these resistant bacteria. Okay, how do we know that just by repeated passage of these diseased tissue states, you're not just creating some form of generic toxin instead of ascribing all this to viral and bacterial causes? So genetic recombination from ultraviolet inactivated bacteriophage particles. You can go through all of these papers, electron microscope studies, reactivation, transfer of self-reproducing units. This sounds promising, right? Because that's what we're supposedly identifying, this reproduction. So they looked again at coli bacteriophages and a host strain of E. coli B, a number of mutants resistant to one another of the bacteriophages is supposedly present. So here, experiments discussed in the paper, ultraviolet inactivated phage particles are still absorbed by sensitive bacteria. Okay, when do we get to the replication mechanism here? The reactivation is caused by some factor in the lysates that have also been supported by the finding. Reactivation occurred with phage that has been purified from extraneous material by differential centrifugation. Okay, so you had to run this through a centrifuge, so you spun it super, super fast, which breaks it down further. And now you're going to make an argument that, again, another process that can't take place inside the human body is getting us closer to the truth. And they do this over and over and over again. If you read this entire paper, you can look through this entire paper, and he makes these assumptions, and he draws these little cartoons, and there's a lot of math that looks very complex and intellectual, okay? There is no point anywhere where he actually identifies a specific mechanism of replication. anywhere in this paper. He's using X-rays and UV light and all this kind of stuff. And he even goes on to say, we also have no evidence as to whether the active units in an inactive particle will proceed to multiply in its single infected cells. The same may be said for the other question, whether the active units remain in spatial relationship to each other. In other words, even in the researchers, much like we saw with Rosalind Franklin, where she said, well, this paper didn't really prove for sure, but we believe it suggests that this, DNA-like structure to viruses. The common narrative is that paper did in fact prove it, even though the author says that it doesn't. And guess what's happening here with Salvador Luria? He's having to admit that we haven't really proved it. And guess what? There's nothing scientific about this entire process. There's no specific mechanism of replication that ever gets identified in any of his work, which is probably why when you read this article and they discuss this, bacterial resistance and genetic structure of virus and the replication mechanism, they're not able to provide a specific citation for it. And I've showed you where you can find all these papers now, all the papers that they cite in this article. He's identified the replication mechanism. Why can't you just concretely say, here's the paper that proves it. They took this specific virus particle and injected it into a human being or into an animal, and we observed replication in living tissue, not in a petri dish, but in living tissue, that this does in fact take place, and via the exact same process as we supposedly isolated it, we were able to get billions more variations and copies of that same thing that we isolated from. They never do that, and it's because they can't, because it's physically impossible. Because all the processes that they engage in order to supposedly produce the results that they're telling you that they get, involve removing genetic material from its natural environment and exposing it to a myriad of different chemicals and physical processes, and then extrapolating out the damage that's done to that tissue as being the effect of some magical virus. Happens over and over and over again.
So now we come to Howard Temin, an American geneticist and virologist. He discovered reverse transcriptase in the 1970s. So this is obviously the element of the viral story that we're interested in, for which he shared the 1975 prize in physiology or medicine with Renato Del Becco and David Baltimore. He was born in Philadelphia, Pennsylvania, to Jewish parents, Annette Lehman, an activist, and Henry Temin, an attorney. As a high school student at Central High School in Philadelphia, he participated in the Jackson's Laboratory Summer Student Program in Bar Harbor, Maine. The director of the program, C.C. Little, told him that Temin was unquestionably the finest scientist of the 57 students who attended the program since the beginning. I can't help but feel this boy is destined to become a really great man in the field of science. Wow, sounds impressive. His parents raised their family to have values associated with social justice and independent thinking, which was evident throughout his life. For Temin's bar mitzvah, the family donated money that would have been spent on the party to a local camp for displaced persons. Temin was also valedictorian of his class, and he devoted his speech to relevant issues at the time, including the recent hydrogen bomb activity and sending a man to the moon. He originally studied embryology, but after about a year and a half, he switched to animal virology. He ran into Harry Rubin, who we've already talked about. He studied the Rous sarcoma virus, a tumor-causing virus that infects chickens. During his research on the virus, he observed that mutations in the virus yielded alterations in the structural characteristics of the infected cells. Now, again, he's using the exact same processes that I've already described in previous lectures that I've already directed you to, so you can go into that in more detail. In 1960, at the McArdle Laboratory for Cancer Research, the University of Wisconsin-Madison recruited Temin as a virologist. Temin stated he was supremely self-confident. When he first arrived in Madison in 1960, he found an unprepared laboratory in the basement of a rundown building with an office that could be considered a closet. He continued his research with RSV at a friend's laboratory at the University of Illinois. While studying Rous sarcoma virus, again, we've already looked at this silliness in the previous lecture, Timmon began to refer the genetic material that the virus induced to the cells, the provirus, using the antibiotic actin-myosin D, which inhibits the expression of DNA. He determined that the provirus was DNA or was located on the cell's DNA. Again, this specific mechanism of action actually hasn't been established, and you're using an antibiotic. It's going to inhibit all sorts of biological processes. The idea that it's just going to be solely unique to DNA to me is silly, but let's continue on here. These results implied that the infecting Rous sarcoma virus was somehow generating complementary double-stranded DNA. Using an antibiotic inhibits the growth, the genetic material in the petri dish. Therefore, they're saying that because of its action on DNA, supposedly, that means that this virus is generating DNA, and they're going to go on to say that this is a this is a virus-producing tumor. Timmon's description of how tumor viruses act on the genetic material of the cell through reverse transcription, so this is the most important part of the Timmon story, this reverse transcription idea was revolutionary. So again, just like with the replication thing, once you say viruses replicate in this way, which was never actually established by Luria, but once you establish that, well, then there's another vector for more drugs that you can produce with more novel mechanisms, which grant you more research dollars. And ultimately, what you'll see is even decades later, none of this has borne any fruit. But I digress. So now with reverse transcription, there's yet another mechanism by which we can use to target drugs that's going to be increasingly genetic, let's say, in nature, which carries us all the way to the COVID vaccines, right? This upset the widely held belief at the time of the popularized version of the central dogma posited by Nobel laureate Francis Crick, one of the co-discoverers of the structure of DNA, along with James Watson and Rosalind Franklin. Crick had claimed only that sequence information cannot flow into DNA or RNA, but he was commonly interpreted as saying that information flows exclusively from DNA to RNA to protein. Many highly respected scientists disregarded his work and declared it impossible. Despite the lack of support in the scientific community, Timmon continued to search for evidence in support of his idea. What do I think is happening here? Individuals above all these people identify what sorts of research could produce results that get them closer toward their goals, namely an increasingly invasive vaccine and increasingly invasive medical systems. And so as they survey the landscape, they suppress ideas until they have had enough opportunity to figure out whether or not they're going to be useful in that agenda and whether or not they can effectively get more money out of said idea. Whether an idea gets promoted or not, or whether an idea gets suppressed or not, has to do with its usefulness in accomplishing certain agendas, its financial capability, and whether or not the environment around said idea is safe enough that they think they can get away with deploying it when it's ultimately not going to be fruitful. And I've referenced this many times over with regard to certain social agendas. They had to wait to aggressively push them until we were sufficiently addicted to the social media, because otherwise you could just unplug with it and the brainwashing would stop. So we're seeing similar concepts here. And again, we see this connection between Rosalind Franklin and James Watson, all of them fitting conveniently into the story that drives us towards specific conclusions. Timmon continued to search for evidence to support his idea, and then working with Satoshi Mizutani, began searching for an enzyme that was responsible for the phenomenon of viral RNA being transferred to proviral DNA. Later that year, Timmon showed that certain tumor viruses carried the ability to reverse the flow of information from RNA back to DNA using reverse transcriptase. Reverse transcriptase was also independently and simultaneously discovered in association with the murine leukemia virus by David Baltimore. And so this is, later, the discovery of reverse transcriptase is important. It's the central enzyme in viral diseases such as AIDS and hepatitis B. So now we've found the mechanism just before AIDS came out for how AIDS works. and it's via this reverse transcription model, even though this increased understanding hasn't yielded much fruit. So once again, I'm not going to go into exceeding detail on these papers, merely to say if you want a deeper understanding, you can reference the lectures that I talked about in the first lecture on this topic. But I will show you the two major papers here. And it's quite interesting how this is presented. So this paper is only 3 pages. And this is the non-Temin paper reviewing this. So RNA-dependent DNA polymerase environs of RNA tumor viruses. What are they doing here? So none of these treatments increase the activity. Incubation at 50 degrees Celsius for 10 minutes totally inactivated the enzyme, as did inclusion of trypsin in the reaction mixture. Addition of as little as 0.01% Triton N100 also markedly depressed activity. So what are they doing here? In order to suppress this replication activity, they're adding trypsin, which I've talked about in previous lectures. They used on COVID because it breaks down cell membranes, so yeah, it's going to inhibit activity. Also something not found in the human body. I also talked about Triton in some of those previous lectures, again, that I showed you in the first lecture. These are all synthetic compounds that are not going to be found in the human body. Then they're using sodium chloride and magnesium acetate and with magnesium acetate, et cetera, et cetera. So what are they doing? They're using chemical compounds like EDTA, Triton, trypsin, et cetera, to suppress the activity of what would, honestly, I would regard as bacterial overgrowth. And they're saying, well, this is capturing inhibition of RNA synthesis. And therefore, this dictates that, well, in order for these bacteria to reproduce, there has to be flow of RNA to DNA. And so by inhibition, using these RNA inhibiting compounds, that must mean that there's backward communication from the RNA to the DNA to cause this replication. Now, what am I saying the replication is? It's not actually replication of viruses, it's just breakdown of these cell tissues that you put inside of a petri dish, and you're just measuring these reactions and ascribing them to viruses, because none of this can take place inside of a human body anyway. That's my position. Again, many lectures I've talked about this in the past, and I directed you to those in the first lecture. So now here we are, and they've jumped to the level where they're saying that this is inhibition of RNA, and because this is inhibiting this process, therefore RNA communicates backward with DNA. None of these concepts, in my opinion, have been proven. I've already showed you some of the issues with this whole Rous sarcoma virus toward the end of the last lecture. So here's the actual Temin paper, RNA-directed DNA synthesis. And this is the basis of reverse transcription, that it goes backward. And this is just a Scientific American article. It's not actually a research paper. He just summarizes findings of research papers that I can't find published anywhere. And he doesn't cite them in the body of his work. So what is he doing here? So he ascribes to viral narrative stuff that I've already shown you why I disagree with in previous lectures. But he winds up using actinomycin D to culture cells producing sarcoma virus. It inhibited the production of all RNA. One would have expected the replication of RNA viral genome to continue without hindrance. So what did he do? He added an antibiotic to the sample. It stopped breaking down to the same extent because he's inhibiting bacterial growth and some levels of cell damage that they're saying are caused by viruses. And he's saying, well, this has a direct action on RNA. Or the reason, once again, that we get this feedback is because we get this inhibition of production of these particles now is because RNA communicates with DNA to produce these particles. So you can disagree with my interpretation or not. The quote-unquote proof for reverse transcription is still the same. They expose a sample to some type of antibiotic or synthetic chemical compound. It stops producing these particles, and they're saying that proves that it's acting on RNA, and RNA reverse feedbacks to DNA. They make that gigantic leap all on their own. And they continue to revise these experiments, but they're still doing this stuff with various serums and synthetic compounds and all sorts of things that cannot take place inside the human body or an animal body. You have to design an experiment in which there's some way to prove that there's this effect taking place in a human body. If you just remove a sample and then just expose it to chemicals over and over again until it produces the result that you want, how can you ascribe all these super specific mechanisms of action to it? But what does this do? It creates a demand for drugs that inhibit reverse transcription. So what does that mean? Just like with the cancer stuff, now we can justify exposing people to toxic compounds like they did with the AIDS crisis when they gave people AZT, a super dangerous drug, because it inhibits viral replication. Oh, look, we stuck it in a petri dish and it was so poisonous it stopped the tissues from proliferating. Therefore, we should give it to our patients. This is the basis of what chemotherapy is based on. And now anything that they say causes reverse transcription, any sort of toxic chemical soup that inhibits this effect in a petri dish, they're going to be justified in giving into patients. And when the patient dies, they blame it on the virus instead of the toxic chemical compound. What an effective, deceptive way to go about poisoning an entire population with an imaginary disease that they don't actually have. Again, I get how controversial this is for people, but go back and watch my virus isolation lectures if you doubt that. And this is all being engineered by one specific group. And now I get to introduce you to Stanley Falco, an American microbiologist and professor of microbiology at Georgetown University, University of Washington, and Stanford University School of Medicine. He is known as the father of the field of molecular microbiology pathogenesis. He formulated molecular Koch's postulates who have guided the study of microbiology determinants of infectious disease since the late 1980s. He spent over 50 years uncovering molecular mechanisms of how bacteria cause disease and how to disarm them. Falco was one of the first scientists to investigate antimicrobial resistance and presented his research extensively to scientific, government, and lay audiences, and how resistance is spread from one organism to another, now known as horizontal gene transfer. So he was born into a Yiddish-speaking household in Albany, New York. His father was a shoe salesman and immigrated from Soviet Kiev, Ukraine. His mother came from a family of Jewish immigrants from Poland. She rented several of their bedrooms and later opened a corset shop. Falco's family was working class to poor, as he described in his memoir. He was therefore a first-generation Jewish American. Falco worked at a toy shop in order to collect enough money to buy a model microscope, as well as admitted to stealing books, one a nurse's book on infectious diseases, in order to explore microbiology as a career path despite his family's financial education. And he was influenced by the book Microbe Hunters, which we saw referenced multiple times already. He went to the University of Maine, finished his PhD thesis at Walter Reed. The two men worked together on isolating mobile genetic elements and transfer of genes from E. coli and Salmonella, etc., etc. But what is his major claim to fame here? His major claim to fame is this molecular Koch's postulate. So what is this? It's experimental criteria that must be satisfied to show that a gene found in a pathogenic microorganism encodes a product that contributes to the disease caused by the pathogen. Sounds nice, right? So what are the three postulates? The phenotype or property under investigation should be associated with a pathogenic member of the species. Specific inactivation genes associated with the suspected virulence trait should lead to a measurable loss in pathogenicity or virulence. Reversion or allelic replacement of the mutated gene could lead to restoration of pathogenicity. So what does this allow us to do? And he's talking, and this is where we start using mRNAs, which of course are later going to be the basis of the COVID vaccines. But what does this allow us to do? Well, it's saying, well, we can find what gene is associated with the pathogenicity of a bacteria or virus, and we should be able to deactivate it. So his molecular postulate gives them access to, well, if we can just turn off this or that gene, if we can just identify this virulent or problematic microorganism or virus, we need to get access to the genetic code of these viruses and of human beings and just figure out which gene it is, target gene therapies at it so we can switch it off. So this is more complex, more highly involved. Again, think about how far we've come at this point, because Edward Jenner's vaccine was supposed to work by literally taking pus from a cow that was infected and injecting it into a person. And they still stick to the story that that worked for smallpox, a supposed viral infection. And now we've come to the point, well, in order to really make a vaccine, we need access to your genetic material and the ability to turn genes on and off. Do you see how crazy this has gotten at this point? If the concept of vaccinations works so well, why do we keep needing all of these revisions and all these different identifications of all these different problems? And we see now that because we've built on the previous faulty assumptions, how ludicrous this has gotten to. And it requires in situ hybridization. So what does this do? We start talking about complementary DNA. Again, I've addressed this in previous lectures. And you get a much more complicated process. It requires that you have precise optimization. You have to preserve the target mRNA, which requires use of formaldehyde and very thin slices. And it has to be embedded in frozen media, which, guess what? Freezing it creates artifacts, which, in my opinion, how can you regard any of this as being accurate? So what does all this require? Increasingly expensive and complex processes that require increasingly specialized training that demand increased funding. What does this create? It creates a money funnel. Anytime you have these increasingly elaborate, more complex technologies that need to be, and how many effective treatments have been generated from all these supposed advances as far as viral replication and all this kind of stuff? Well, as far as American society goes, after we started getting indoor toilets and all that kind of stuff, indoor plumbing, our health improved dramatically. But since then, in the last, I would say, definitely 60 years, we've gotten sicker and sicker and sicker. So where has all this advanced technology gotten us? Look at the actual outcomes. I get that this sounds very complicated and very intellectual, and you're probably having a hard time, following everything that I'm saying. I get that. If you're not familiar with this stuff, it's very difficult to follow. But just ask yourself, where has all this technology, all these billions of dollars, all these advanced techniques that they developed, in terms of results, where has this actually gotten? Because if you're watching this video, you know they lied about the effectiveness of the COVID shot. You know the damage is worse than that the cure was worse than the disease as it relates to the COVID stuff, if you've been paying attention to all-cause mortality. This is where this style of thinking gets us, where you make massive logical leaps that are not axiomatic. That none of this in any of these viral papers would you make the argument that, okay, from this result, then this must be the next conclusion. That's an axiom. Nothing is like this. They make gigantic leaps from examining the effects of what's happening in a petri dish when you expose it to a certain chemical compound. And Mr. Falco, in line with the Jewish tradition we've seen thus far, has advanced this exact same agenda with this concept of a molecular Koch's postulate, which, by the way, none of these viruses have ever fulfilled Koch's postulates. They say that that's outdated now.
So here is where I start discussing in detail the compounding problems with this isolation procedure as it relates to SARS-CoV-2, and we keep using more and more advanced technologies to supposedly isolate these viruses. But again, I traced it back to the beginning of virology. We've never once ever isolated a single virus. That's never happened. We've observed the effects of filtrates on other living things, and we've observed what happens when you expose a tissue sample to things like trypsin and fetal bovine serum and other chemical compounds that have known significant effects, and then observed the products of that and then referred to them as viruses. But here is where I discuss more of that in detail. Here's where I discuss the cDNA, the fact that all these genomes that they're reporting in these virus researchers, we know that there's problems with reproducing those genomes using the exact same procedures. So even if you're going to argue with me that these primers are somehow accurate in detecting these viruses, whenever you go back from scratch and try to do that stuff again, we're not able to reproduce those genomes. So as far as I'm concerned, you just take an amalgamation of genetic material, separate it out into constituent elements, then you plug that into a computer program so it spit out a sequence, and now every time that you go back to test for something, if the amalgamated DNA that you've gathered matches with your test, congratulations, you have a quote-unquote virus. Despite all the technical terms that they use, etc., this is ultimately what you're doing, because again, if you go back to the Jenner lectures, you never proved the concept of your virus in the first place. So here I discuss problems with using PCR and this concept of cytopathic effect that's often cited. I continue to talk about virus isolation, this concept of exosomes, and the fact that we're identifying these structures when they're being treated with penicillin and all of these structures, and the fact that a lot of the things that they identified as SARS-CoV-2 are structurally identical to pseudopods identified in the kidneys far before we ever heard about anything as it relates to COVID or SARS or any of that stuff. And here is where we first touched on the works of Paul Ehrlich, which brings us to Walter Ian Lipkin, John Snow Professor of Epidemiology at the Mailman School of Public Health at Columbia University and Professor of Neurology and Pathology in the College of Physicians and Surgeons at Columbia University. He is also Director of the Center for Infection and Immunity, an academic laboratory for microbe hunting in acute and chronic diseases. Lipkin is internationally recognized for his work with the West Nile virus, SARS, and COVID-19. Now, I've touched on all of these in previous lectures. Again, I've showed you where you can find additional information about the farce that is the COVID-19 quote-unquote epidemic. But here we go. Let's see what Wikipedia says, and then we'll provide some clarification. Lipkin was born in Chicago, Illinois, where he attended the University of Chicago Laboratory School and was president of the student board in 1969, relocated to New York and earned his BA from Sarah Lawrence College. He felt that if I went straight into cultural anthropology after college, I'd be a parasite and have nothing to offer, so I decided to become a medical anthropologist to bring back traditional medicines. Suddenly, I found myself in medical school. He became a clinical clerk at the UCL Institute of Neurology at Queen Square, London on fellowship, and an intern at the Medicine of University of Pittsburgh. He completed a residency in the University of Washington and completed a residency in neurology in the University of California, San Francisco. He conducted postdoctoral research in microbiology and neuroscience at the Scripps Research Center from 1984 to 1990. Under the mentorship of Michael Oldstone and Floyd Bloom, in his 6 years at Scripps, Lipkin became a senior research associate. So allow me to add a little bit of additional information that was left out of the Wikipedia article. One of 3 children, his parents were of Russian Jewish background, met at the New School in New York City. This is sciencehistory.org, by the way. You can see the URL here. But moved to Chicago to study at the University of Chicago. His father is of psychology and his mother of social work. So they mention him going to Rush University and the Neurology Institute of London. They also say he spent some time at Indian Health of Oklahoma, and they go on to say this stuff about University of Washington, but he's attending the same universities. He also became interested in AIDS. His next stop was Scripps Research in La Jolla, California, at fellowship at Michael Oldstone's lab. So we have Floyd E. Bloom, an American medical researcher specializing in chemical neuroanatomy, received A.B. cum laude from Southern Methodist University in 1956, chairman of the Department of Neuropharmacology at Scripps Research Institute in La Jolla, California, director of behavioral neurobiology at the Salk Institute. Yes, we've already touched on Jonas Salk, chief laboratory of neuropharmacology in the National Institutes of Health. He is the member of the National Academy of Sciences and the American Philosophical Society. Additionally, left out of the Wikipedia article, he spent 2 years as an intern and resident at the Barnes Jewish Hospital. So take from that what you will. His other mentor, Michael Oldstone, that we see here, has a biography on the NCBI database. Recently elected member of the National Academy of Sciences, has devoted his career to the molecular understanding of how viruses infect cells, evade and manipulate the immune system, and cause diseases. Once again, these are supposedly inert genetic sequences that have been endowed with intelligence somehow, but I digress. He studied the pathogenesis of lymphocytic choriomeningitis, the mouse, for decades. He overturned a deeply entrenched dogma about immunological tolerance and made numerous observations that shaped fundamental concepts in virology, immunology, and infectious disease. So here's Mr. Oldstone, born in Manhattan, junior high school, devoured the 1926 classic book, Microbe Hunters. It's interesting how influential this book was in shaping all of these virologists and how that individual was shaped by Paul Ehrlich's vision as it relates to this stuff. And he was educated by Theodore Woodward and again, highly influenced by the Microbe Hunters book. Oldstone was invited to spend the summer of his third year of medical school working at Beth Israel Hospital in Boston, Massachusetts, in the laboratory of Jacob Fine, one of the world's leading researchers on endotoxic shock. We'll go on through this here, and of course, two of Mr. Oldstone's primary mentors are Sheldon Greisman and Jacob Fine here. And of course, unable to find their ancestry, but we know that their last name Greisman, often a Jewish last name. Here we have Mendel Greisman. And then we have another Jacob Fine, not the same one, who is, of course, a rabbi. So we have someone working hand in hand with somebody who's Jewish. We have Ian Lipkin himself, who's Jewish. And then his other mentor, Michael Oldstone, who both of his mentors are likely Jewish. So let's return to Mr. Lipkin here. He has a reputation as a master virus hunter due to his speed and innovative methods of identifying new viruses, which have been lauded by the National Institute of Allergy and Infectious Disease Director Anthony Fauci. Lipkin has also advised the Chinese government and the World Health Organization during the 2002-2004 SARS outbreak. Now, I've touched on that in previous lectures. It's been over a year since I've done so, but those things were utter failures. And again, the questions during the SARS outbreak and the 2009 outbreaks and all these outbreaks have been, why did we rush to such an aggressive response and even in some cases generating vaccines that were entirely unnecessary and unsafe? So I've talked about that in some detail previously. But he's the guy that's telling China and the World Health Organization what your response should be. He is the Director for Research and Diagnostic Discovery under the National Institutes of Health and Centers for Excellence of the Translational Research Program. And he also served as a science consultant for the film Contagion. It has been praised for its scientific accuracy. So you may have seen clips of the movie Contagion whenever the COVID outbreak first happened, and people were freaking out about the concept of, look, they're saying it came from bats, and look, and it came out of China, and look, it's bats, and it came out of China in the movie. Isn't that interesting? So this same guy who's working along this same lineage is the one who's consulting on the film Contagion. He also was a key part of the AIDS crisis epidemic. Again, you can go back and watch my homosexual lectures if you're so interested. Lipkin was the first to identify a microbe using purely molecular tool. Here we see two papers, and I don't want to go into full detail on this because it's going to be another 4 hours of discussion. But you can go back to my previous lectures and apply this same concept where I talk about the idea of cDNA being not very scientifically robust. And here he's arguing for it as being a key agent in this Bornavirus, and he's the first one to identify it. And so we see in 1989 with Lipkin and his tool that we're about to look at, there's an explosion of identification of this virus and that virus and this virus and that virus. And I just find it so interesting that after polio, there's an escalation of all these viral ideas, obviously. And the next major occurrence is the AIDS crisis. But after the AIDS crisis, you see this almost stepwise progression of viral outbreak to viral outbreak. So you see Bornavirus, then West Nile virus, then SARS-CoV in the early 2000s, then MERS-CoV, then myalgic encephalitis / chronic fatigue syndrome, then acute flaccid myelitis, then SARS-CoV-2. So you have these intervening periods in between this time frame where you have sort of Ebola and the bird flu and et cetera, that were all kind of happening sequentially. But Mr. Lipkin in 1989 kicked off the molecular tool phenomenon. Now, as it relates to his main push for this mass identification of all these new supposed viruses, he uses something called mass tag PCR. So it all comes back to what? It all comes back to the first main lecture that I ever published. This whole viral scenario, this whole trajectory that we've been on, it all comes back around to this stupid test. It's based on mass spectrometric detection of an end product. This technology was developed by researchers from a Mailman School of Public Health at Columbia University and the Columbia Genome Center. Likely, like conventional PCR, mass tag PCR uses primer pairs. The difference in primer pairs used for mass tag PCR are tagged with molecules of known masses or mass codes. Instead of a single pair of primers, this technology uses a number of primers, making it a multiplex system. Unlike conventional multiplex PCR, in mass tag PCR, more than 15 primer pairs could be used. If DNA from any of the agent of the primer panel is present, it will be amplified. Each amplified product will carry its specific mass codes. The PCR product is then purified to remove unbound primers, DNTPs, enzymes, and other impurities. Finally, the purified products are subjected to UV light at the chemical bond between the nucleic acids and primers is photolabile. As the mass codes are liberated from PCR products, they are detected with a mass spectrometer. Technique has been found to detect previously uncharacterized clade of rhinoviruses. It's more comprehensive and a more sensitive technique. Despite the fact here that they're asserting that there's more primer pairs, if this test is more sensitive, and we don't have the protocols for this, by the way, and we know that even if you're agreeing with the accuracy of the primer pairs, which I don't, the argument that they're going to use here is that, well, there's multiple primer pairs, but it's more sensitive. But we know that even if you agreed that the primer pairs were actually accurate, which once again I don't, at 35 cycles, there's a 97% plus chance that it's a false positive. So if we don't have access to the protocol that you're using for this mass tag PCR, you could be identifying all sorts of viruses all the time. And that's a big part of what they've been using with this. We found more COVID in the water, we might need to be able to do lockdown. And it all circles back to this Mr. Ian Lipkin, who criticized governments and is advising the Chinese government, and he himself is Jewish, his mentorship is Jewish, and he accurately predicted the COVID scenario 9 years ahead of when it actually was going to happen. Also, here's my first discussion where we looked at the NIH itself and Anthony Fauci, but specifically his role with using AZT as a treatment for AIDS. So that's where this reference, October 24, 2021, that's where you could find some more information on that. This was a repurposed cancer drug that produces the exact same side effects with it that were ascribed to AIDS. And there's many people that have done work on a lot of the recreational drugs that were used in the gay community that can lead to, immune system failure and things like that that are going on in this specific population. So again, take from that what you will. What about AIDS? Well, you have Willie Rosenbaum, co-discoverer of the human immunodeficiency virus, along with Jean-Claude Chairman of Luc Montagnier's team. He has, since November 19th, 2003, held the chair of France's Conseil Nation du Sida, and before that, had since 1989 practiced in infectious and tropical diseases. So, where did this come about? It came about from a Lancet article in which he was the lead author in 1982 titled Multiple Opportunistic Infections in a Male Homosexual in France. That's it. And then by 1984, they had isolated a retrovirus for this immune deficiency syndrome, and then it exploded after that. Not listed here is his ancestry, which, as you can guess, based on his last name. He's listed in here French people of Polish-Jewish descent, Willie Rosenbaum, and there's his picture. Interestingly, not listed in the Wikipedia article itself. So here is that first paper that was found by Mr. Rosenbaum. Multiple opportunistic infection in a male homosexual in France. The first cases of acquired cellular immunodepression among male homosexuals were reported by the Centers of Disease Control. Since then, several cases of opportunistic infection and of Kaposi sarcoma among male homosexuals have been reported in California and east of the USA. In Britain, a case in a homosexual who had been to the USA had also been reported. The patient is a male homosexual, age 38. His last visit to New York was in February 1980, when for the first and only time he took poppers several times. He has never taken other drugs before or since. He was referred to the Claude Bernard Hospital in July 1981 because of continuous high temperatures, loss of 5 kilograms in weight, malaise, and dry cough. Clinical examinations revealed disseminated and microlymphadenopathies only. The chest x-ray was normal. The white blood cell count was 4,500 microliters, 47% neutrophils. He's talking about white blood cell types here. 10% eosinophils, 33% lymphocytes, 10% monocytes, without abnormalities. Levels of his antibodies, which we know the problems with the antibody stuff. IgG and IgA were high. IgM levels were normal. Circulating immune complexes were present. Specific IgM antibodies were absent. There were no HB antigenemia, but the antibodies to HBsAg and HBcAg were present. This time the chest x-ray showed interstitial infiltration. At the end of August, a left hemiplegia appeared. He started to get paralysis of his left side. Oral thrush was observed. Bronchiolar lavage revealed numerous pneumocystis carinii cysts. Cotrimoxazole was given, whereupon the clinical chest x-ray and neurological findings improved, suggesting a brain nocardiosis. Biopsy indicated abnormal immune response similar to that observed by angioimmunoblastic lymphadenopathy. At this time, the antibody levels had increased. He had pleuropneumonia caused by Mycobacterium tuberculosis, and specific treatment was instituted. For the first time, CMV was detected in the blood and urine and is still present. Okay, so we see this individual case, this individual outbreak here, and all these immune markers. But he says he took poppers several times. He took them for the first and only time, but he took them several times. So isn't that interesting? Now, the thing about addicts is that they lie. So I don't know that I trust that this was the only time that he did this. But what are poppers? Popper is a slang term given broadly to recreational drugs of the chemical class called alkyl nitrites. They are inhaled.
They act on the body as vasodilators. Most widely sold products include the original isoamyl nitrite, isopentyl nitrite, and isopropyl nitrite. Isobutyl nitrite is also widely used but is banned in the European Union. Poppers are labeled as packaged as room deodorizers, leather polish, nail polish remover, or videotape head cleaner to evade anti-drug laws. They cause a relaxation effect on involuntary smooth muscles, such as those in the throat and anus. It is used for practical service purposes to facilitate anal sex by increasing blood flow and relaxing sphincter muscles. This drug is also used for recreational purposes, typically for the high or the rush that the drug can create and enhance sexual pleasure in general. Poppers are part of club culture from the mid-1970s disco scene and surged in popularity in the 1980s and 90s. And given the description of this, what group is most likely to be taking this type of recreational drug? Involuntary smooth muscle relaxation. Yeah, we know what group's most likely to take use of this drug. And they say, it was reported among gay men, but quickly spread to avant-garde heterosexuals. So they jump to that, and then they show this chemical structures, and it has this toxicity effect. Reports insignificant hazard associated with inhalation of alkyl nitrites. British government guidance relative to harmfulness places them in the less harmful recreational category. Swallowing may cause cyanosis, methylglobinemia, unconsciousness, coma, and complications leading to death. And accidental aspiration may cause lipoid pneumonia. They may cause maculopathy or eye damage. Some studies concluded may be increased risk of retinal damage. So they talk about eye stuff, eye surgery, and that it also has carcinogenic properties, and it's possibly carcinogenic to humans. So this is all that they say about this. They talk about legislation and blah, blah, blah. Okay. This is health hazards of nitrite inhalant. So notice how they didn't say anything about the immune system whatsoever. And they said, well, if you swallow it, it can be a problem. But inhaling it is not that dangerous. But this is 1988. So we knew this just a few years after. And you can see the individuals that put this report together. The use of drugs may expedite disease progression by decreasing immune function and acting as a vehicle of transmission for other infectious agents. So they're saying that they're concerned because AIDS patients, this might worsen their AIDS because it causes the exact same things that AIDS does. Drug abuse may promote the development of malignancies. Nitrite inhalants, commonly used by homosexual men, have been associated with Kaposi sarcoma in AIDS. The mechanism of action of nitrites as a cofactor, if any, has yet to be elucidated, because I don't think they want to know. Toxicological effects of various forms of alkyl nitrates, the effects of nitrate use on the immune system, and epidemiological findings associated with nitrite use with KS and AIDS. The purpose of this introduction is to provide an overview of issues and controversies raised, covered in more detail in individual chapters. Besides participating in the formation of carcinogenic metabolites, so as we'll see, this Kaposi sarcoma is associated with quote-unquote AIDS, and guess what? This thing that they said might be a carcinogen, in the Wikipedia article they're saying it forms the metabolites that contribute to cancer. They may increase the likelihood of Kaposi sarcoma by altering immune function. They studied the effects on immunological function using two strategies. First, examining effects on lymphocyte number and thymus weight. Second, measure a pre-treatment and response of immune components to the individual with various adjuvants and antigens. Given the complexity of the immune system, the possible variations of parameters, a large number of dependent variables and sampling times were chosen from. So they said Lewis reported no significant detrimental effects. However, two other researchers showed different patterns: decreased T lymphocyte numbers, which is exactly what's associated with HIV and AIDS, and changes in immune function after 21 weeks of intranasal amyl nitrate in mice, and after 13 sessions of amyl nitrate inhalation in human volunteers. So we know that this is the group that's consuming these compounds, and we know that these compounds contribute to decreased immune function, but what are they saying? Well, it might enhance the AIDS. How come we don't know that this isn't the entire cause? Much has been written about the extent of nitrite use among homosexual men, but these surveys are not distinguished by use of sexual orientation. Okay, and they say it may act as a cofactor for AIDS. Godert found that suppression ratio of lymphocytes was lower among users of volatile nitrite than non-users. First indication of the potential to suppress the immune system, the possibility that poppers were only a surrogate that were highly correlated with popper use was not ruled out. So what? They never actually determined for sure, hey, let's do individual studies where we're drug testing these guys to make sure that they're not using poppers. Cellular cytotoxicity was demonstrated, as was depression of natural killer cells, which is also associated with AIDS. We suggested that continuous use of these substances might interfere with attempts to correct the immune system by other agents, and therefore their use should be condemned. But now what happens? If you show up and you have a depreciated immune system, what are they going to do? They're going to diagnose you with HIV, and they're going to put you on AZT, which is a toxic cancer drug, because it kills this stuff in vitro, because it's a poison, but it also suppresses your immune system. So just like with COVID, I talked about environmental exposures that contribute to the shortness of breath, et cetera, et cetera. You can go back and watch the 5G lectures if you doubt me on that. And then I talked about the remdesivir, which contributes to the systemic system symptoms that they associated with COVID. It's the exact same playbook all over again. And here's the actual supposed isolation of the virus. Again, if you've witnessed my virus isolation lectures already, I've covered this in more detail, but you're going to see the exact same process they do again. They're going to use antibiotics on the sample. They're going to use an electron microscope image from a still stained image. And they're going to say, look, these broken down particles are AIDS virus. Again, I've critiqued all these things in the past. I've kind of showed you where you can find those lectures. I get that this is a lot of information. So they add the fetal calf serum and the antibiotics, and yet they still are never able to take what they've supposedly isolated, inject it straight into another animal, and make them sick. It never works. They never are able to reproduce experiments like that. It's only in petri dishes by looking at breakdown products of these tissues. They can't ever replicate the disease conditions that they're saying that they do. So we report here the isolation of a new retrovirus from the lymph node of a homosexual patient with multiple lymphadenopathy. The retrovirus was propagated in cultures of T lymphocytes from a healthy adult donor and from the umbilical cord blood of newborn infants. So that sounds promising, right? Hey, we took a healthy adult donor sample and we got these results. Except for what? They cultured it out separately on a petri dish that's removed from the human's body, so it's now in an unhealthy environment. So again, you have to do a control experiment where you don't expose any of this process to what you consider to be the virus. So you take the healthy adult donor, you don't expose it to any of this supposed virus, you just run all the other processes on it, and you see if it results in the exact same thing. And in all the research stuff that I showed you back in my virus isolation lectures, the end result is the same, even if you don't expose it to the supposed virus, because the process that you're doing causes the breakdown of the tissue. So viral core proteins were not immunologically related to these proteins, it says. 15 days after culturing the T cells from the lymph node, a retrovirus could be detected in the cell-free supernatant by its reverse transcriptase activity. So what is the reverse transcriptase activity? Well, if we can tell, because when we don't kill the tissue by exposing it to some compound that we say acts on RNA, or we say acts on reverse transcription, it continues to produce particles. Therefore, we know it's acting on reverse transcription. These are the logical leaps that they're engaged in. The production of the virus increased for 2 more weeks. This rise was followed by a decline in both viable cell count and virus production in the culture. So when they say virus, what are they talking about? They're talking about when they zoom in on the cell, they're saying, look, there was more of these blobs that formed. Some people call them exosomes as well, and then people will, look, it's budding off of the membrane. How do you know that if this is just a still cross-sectional picture? that you've treated with staining materials and all this other stuff? How do you know that you haven't just dehydrated and degenerated this membrane and these particles just broke off as a result of that, and they're just basically congealed tissue coming off of here and have nothing to do with independent particles that are from some virus themselves? This is the proof that they're appealing to. These little dots. We've isolated the virus because look, when we zoomed in and treated this thing, these little dots form. To date, we have been able to propagate our virus isolate only in mature T lymphocytes. That's table 2. So in other words, they tried it on a whole host of other tissues, which, by the way, we knew from the multiple opportunistic infection thing that this is supposed to act on natural killer cells too. And guess what? We've seen in the health hazards thing on the nitrites that it does in fact act on natural killer cells and all other types of white blood cells. But here, they're only able to get this process to work whenever they have a full lab to do it in, only in mature T lymphocytes. So what did they do? They added antibiotics, which are anti to life. Okay, they added 10% fetal calf serum, just like they did in all the other virus isolation nonsense. Then they added interferon. So I broke down, by the way, the effects that these antibiotics and stuff have on the human body and human tissues in those virus isolation lectures. So again, if I understand that this is a lot of information, but you can go back and check that. And then they added Triton, once again, just like all the other stuff. Assays were performed with disrupted virus in final volume and diethoritol. And they added magnesium chloride. They add all these other additional things that you wouldn't take place inside the human body. They couldn't get immature T cells to be infected with the virus. They couldn't get B lymphoblastoid cell lines to be, quote unquote, infected with the virus. All attempts to infect all these other types of cells were unsuccessful. So what do? They just keep tinkering with biological tissue until it spits out a result that they feel like they can use. And then they say, look, we isolated a virus. Even though they can't ever really grab this particle. So this particle is supposed to be a virus that can replicate itself and hijack the machinery of the human body. They can't ever just like snag one of these and put it in another body and then just have it proliferate. Because even in this mature T lymphocyte that they're talking about here in table 2, they take this tissue sample and they put it in a petri dish and they add all these other chemicals to it to get it to break down and form these particles that they call virus. Immature, premature T cells, etc., etc. The viruses have in common high tropism for helper T lymphocytes and possess a reverse transcriptase, which is magnesium dependent. So what? They're saying that, it's reverse transcription, so it hijacks your genetic machinery and gets it to produce these viral particles that normally produce your own genetic code. It hijacks this to produce its genetic code, even though it's this inert segment of genetic material that doesn't have any intelligence associated with it. But we have to facilitate this process by giving it a bunch of magnesium. Again, excess levels of magnesium also have toxicity associated with them, and if you don't have the products that absorb and break down magnesium, it can cause all sorts of problems. So look, they tell you how much magnesium and manganese and all this kind of stuff they had to incorporate in it, again, to get this entire process to work. Cells were treated with polybrine and then infected either with co-culture virus-producing cells or directly by virus-containing supernatant. Supernatants were harvested 1 to 2 weeks. So here's the deal with stuff like this. And then, of course, there was a huge campaign for a vaccine, which they've never had a successful vaccine for this stuff. Okay, if magnesium is dependent for this reaction, then how come some sort of magnesium inhibitor, selective magnesium uptake inhibitor of some kind, couldn't be used as an effective treatment? Because you just said, well, this doesn't work without the magnesium. This doesn't work without a bunch of antibiotics, too, and the fetal calf serum and the petri dish. But if this is your proposed mechanism, and you're going to go after the reverse transcription part, wouldn't it be far easier and far simpler to find something that stops the magnesium part of the reaction? Of course it would, but it would be a less complex drug, far easier to design, and also when it failed, because when these homosexual men kept taking poppers and kept getting sick, and this didn't make them better, you'd be up a creek. So what did you do instead? You gave them a highly dangerous cancer drug, AZT, which also suppresses the immune system to supposedly kill the virus, and you retroact and you take them on and off of it, and eventually their immune system gets so suppressed from all those different cycles of using those drugs that what happens? They get pneumonia or some other sort of opportunistic infection and they die from a problem that was caused from a reckless lifestyle. We're coming full circle from the homosexuality lectures that I covered as well here. This is their point of proof. Regardless of how you feel about my interpretation, this supposedly proves that this individual particle crashes gay men's immune systems and causes all these symptoms, which we also know can be caused by the poppers that the homosexual community was known to be taking. Very, very crafty on Rosenbaum's. And when we look at this Spanish flu, this figure of Jeffrey K. Taubenberger emerges, an American virologist. With Ann Reed, he was the first to sequence the genome of the influenza virus, which caused the 1918 pandemic of Spanish flu. He is chief of the Viral Pathogenesis and Evolution Section, Laboratory of Infectious Diseases, National Institute of Allergy and Infectious Diseases at the NIH. His laboratory studies a number of viruses, including influenza A viruses, which are the pathogens that cause yearly flu epidemics and have caused periodic pandemics, such as the 1968 outbreak that killed an estimated 1 million people. So who's doing the surveillance? Who's doing the every year you need to get your flu shot type stuff? Well, one such figure is, of course, Jeffrey Taubenberg. His research aims to inform public health strategies on several important aspects of the flu: seasonal flu, avian flu, which circulates among birds and has infected humans in the past, swine flu, which circulates among pigs and has infected humans in the past, and pandemic flu, which can arise from numerous sources because humans have little to no immunity to it. Taubenberger was born in Germany, the third son of an army officer. When he was 9, he moved to a suburb of Washington, D.C. with his parents after his father was posted at the Pentagon. He completed a combined MD and PhD at the Medical College of Virginia in Richmond in a course designed for students who wanted to follow a career in medical research. For his thesis, he studied how stem cells of the bone marrow differentiate into mature cells of white blood cell systems. In 1988, he began a training to become a pathologist at the National Cancer Institute of the NIH. In 1993, he was recruited to the new lab at the Armed Forces Institute of Pathology. He was one of more than a dozen tenant facilities located on the campus of Walter Reed Army Medical Center in the northeast of Washington. So he started looking at bottlenose dolphins, and then here, in search of the 1918 Spanish flu virus, fearing government cutbacks, so he was afraid that he could potentially lose his position, he looked for an application of PCR to the immense warehouse of tissue samples at the AIFP. So he was doing this out of desperation. He eventually settled on finding the remains of the flu virus, which caused the 1918 Spanish flu. Taubenberger's team searched for samples of victims who had succumbed to the initial viral infection and not the subsequent bacterial pneumonia. 7 samples seemed promising. So he's looking at people that have been dead for 80 years, and he's examining their samples. Genome of the flu virus includes about 13,000 base pairs, which is decayed into small pieces as small as 100 base pairs. Primers have to be constructed for PCR to work, a concept I'm very familiar with. They bind to the fragment, and with the help of the polymerase bases are added between the primers to make a copy. Millions of copies of the gene segment are labeled with a radioactive probe, etc., etc. So I showed you where to find this in more detail because I'm about to go after his paper. And then a novelist comes along and discovers Taubenberger's paper through an interview in Science and wanted to know whether Taubenberger would be interested in reading a draft of his novel. So just like we've already seen with the virus hunters thing that came out, that was largely influenced by the work of Paul Ehrlich, now we have this same thing that's being pushed out into the zeitgeist, right? He then agreed to be interviewed by Carter about his ongoing work, and which would lead to the 1918 strain ultimately being reconstructed. Wow. Okay. So that's the story. So nothing about Taubenberger's ancestry is listed in here. However, I have Torah Cafe. Here's Dr. Jeffrey Taubenberger, an American virologist, chief of viral pathogenesis and evolution section, Laboratory of Infectious Disease, National Institute of Allergy and Infectious Diseases, National Institute of Health. Confronting moral dilemmas amid a pandemic. And here he is with Rabbi Dr. Edward Reichman. And this lecture took place at the 10th Annual National Jewish Retreat. So at the 10th Annual National Jewish Retreat, Mr. Taubenberger is delivering this morality of behavior during a pandemic type scenario, and this took place in 2015. So here is his paper. This is how he returned and erected the narrative as it relates to the Spanish flu, and how he found out that it was actually H1N1, that the Spanish flu was actually H1N1, and he used PCR to do it. So I'm just going to highlight a couple of key things. Now, again, in order for my conclusions to be acceptable, if you're not familiar with the PCR lectures, I showed you where to find them, but they're critical for an understanding of this paper. But I'm going to point out where the flaw is in the reasoning, and you can go back and look at those lectures if you doubt my assertions here. So, initial genetic characterization of the 1918 Spanish influenza virus. The Spanish influenza pandemic killed at least 20 million people. Now notice how it's progressed to between 50 and 100 million is the projected number today. Okay, so in the 90s, we were saying it was 20 million. The farther away we get from it, the more people it killed. Understanding the origins of the 1918 virus and the basis for its exceptional virulence may aid in the prediction of future influenza pandemics. RNA from a victim of a 1918 pandemic was isolated from a formalin-fixed, paraffin-embedded lung tissue sample. What does all that mean? They preserved it in a form of wax, essentially, for 80 years at this point. 9 fragments of the viral RNA were sequenced from coding regions of the hemagglutinin neuroaminidase nucleoprotein matrix protein 1 and 2. Because pigs can be infected with both avian and human strains, they are thought to be an intermediary in this process. Analysis of survivor antibody titers from the late 1930s and historical projection of the phylogenetic analysis, blah, 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 suggests the 1918 strain was an H1N1 subtype. Now, I've already addressed the antibody problem, the origins of the ideas of antibodies, the fact that a lot of the visualizations they say are antibodies are structurally identical to other things. So, again, using this retroactively as proof to me is asinine, but that's not what I'm going to appeal to disprove Mr. Taubenberger's process here. So they took this tissue from U.S. servicemen killed in the 1918 pandemic. We randomly selected 28 cases for pathological review. The majority died from bacterial pneumonia, one of the most common sequelae of the pandemic. So in other words, this is the Fauci thesis put forward, basically, that most of the people that were dying during this time frame with respiratory problems died from bacterial pneumonia, not from viral infection directly, and because they were finding, bacterial stuff on masks. Again, my position with regard to the bacterial stuff is bacteria on its own is normal, healthy, and natural. You have to change something about the environment nutritionally, be in an unclean situation like, trench warfare or something, in order for bacteria to become problematic. Bacteria in and of themselves are not pathological on their own. They have to be exposed to some sort of unhealthy environment. But again, you don't have to accept that premise. We'll just continue on. We chose 14 formalin-fixed paraffin-embedded tissue specimen from 7 individuals for further analysis. 3 had bronchopneumonia, and 1 case showing asynchrony between the disease in the left and right lungs. This case showed acute bacterial lobar pneumonia in the left lung, but focal acute bronchiolitis and alveolitis in the right lung, which is indicative of primary viral pneumonia. So all of this is based on what? They preserved these people's lungs in wax for 80 years. They cracked them open, looked at the histological or tissue quality, and said, well, all these other lungs look filled up with pneumonia, but this one has one lung that's filled up with pneumonia, and this other side looks like it's inflamed. Therefore, that's quote-unquote indicative of viral infection. So we're saying that, therefore, that means that this definitely has the Spanish flu in it. This was the only case among the 28 in which the features of early viral pneumonia was conclusively present. So of all the samples they had, coincidentally, just one had the unique presentation necessary to justify saying, well, this is viral. So what did they do? Control amplification of the reverse transcribed fragments of the PCR for B-actin was positive in 11 of 14 formalin-fixed paraffin-embedded tissue samples examined. However, RNA templates larger than 200 were not amplifiable. They had to use smaller sequences. We therefore designed 9 degenerate consensus RT-PCR primer sets to amplify small fragments. Under 200 primer set of genes encoding agglutinin, neuroaminidase, nucleoprotein, 2 fragments, etc., 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 and 1 human. Let me pause that for a second in case you missed it. They designed the primer sets that they're comparing this to. Do you see what I'm saying? They synthetically designed these primers that they're going to test against this sample. So they could constitute these primers any way that they want to in order to get the results that they want. So later on, they're going to say when they compare these primers, oh, well, it's this is the only one that matched up. Yes, because you synthetically designed the primer to match up with it. But again, this is not the point of proof that I'm going to appeal to. No other influenza strains had been used in this laboratory. We were able to amplify and sequence all 9 fragments of the virus from 1918 case 1. Again, how do you know your genetic material is virus? But we'll continue on. With different tissue blocks, none of the other cases selected by histological criteria and examined for influenza virus RNA were positive. Although the length of the sequence between primers was small, phylogenetic analysis for each segment were possible with the use of corresponding regions of other previously sequenced influenza viruses. So now they're stitching this together just like they did with COVID, which I talked about back 2 years ago. They're stitching these sequences together synthetically to make this whole in this process. So they designed these primer pairs. Again, designed means they created the primer pairs. They're not being generated organically. But just before this gets too convoluted and I try and poke too many holes that I don't have enough time to point out the reasons why. Here's their protocol for the PCR test. It's at this point, again, I showed you where you can find those PCR lectures. Whenever you're generating a PCR test, there are manuals and books that tell you the standardized procedure for developing said test. In order to make this accurate to the extent that it can be. Now, before you even get to the PCR, based on stuff that I've talked about previously, there's massive problems with this virus isolation process to begin with. They're using fetal calf serum and exposing it to all these synthetic compounds and trypsins and antibiotics and all this kind of stuff that degenerates the sample, and then they call that the cytopathic effect, and that it's caused by this virus, and then they observe the degenerated structures that they've just poisoned, and then they say, look, these particles are viruses. And then they say that it's replicating because because the tissue's breaking down. Established all that in previous lectures. Before you even get to the PCR, all those problems are present. But just highlighting the PCR here, you are supposed to create a PCR with an annealing temperature that is close in all phases that you're executing it, meaning it needs to be within a couple of degrees Celsius in the phases in which you're doing this test. And the cycle threshold, which is the part most people are familiar with the PCR test, matters. So, for example, Anthony Fauci himself said, if you are testing a sample at 35 cycles or above, the odds of it being replication competent are minuscule. And just as a refresher, the cycle threshold works on a logarithmic scale. All that means is if you go from a cycle threshold of 3 to a cycle threshold of 4, it's twice as sensitive. If you go from a cycle threshold of 3 to 5, it's 4 times as sensitive. So going from a cycle threshold of 35 to a cycle threshold of 36 is twice as sensitive, and the results are that much more meaningless. So what protocol did they do for the PCR test here to identify the Spanish flu stuff? That became H1N1. And the H1N1 narrative started largely in the early 2000s about how dangerous H1N1 is. Well, their reverse transcription was carried out at 35 degrees Celsius for 45 minutes. And then they break down how they use this leukemia virus. So they're having to put this in, and then they show you the 200 nanometer length and 10 millimole depth, which is an inappropriate length for these sequences, but we don't have to address all that. I'll say, okay, so now cycling conditions were as follows. 9 minutes at 94 degrees Celsius. At 40 cycles is how they executed their PCR test of 94 degrees Celsius for 30 seconds. 50 degrees Celsius, which are at 45 degrees Celsius for 32 seconds, and 72 degrees Celsius for 30 seconds, then 5 minutes at 72 degrees Celsius. 1/6 of the reaction was separated on a 7% denaturating polyacrylamide gel, then dried and visualized, etc., etc. So what did they do here? The temperature fluctuates wildly from 94 to 50 to 45 to 72. This makes the results entirely inaccurate and prone to false positives. And they'd crank the cycle threshold up to 40 cycles. So what did Mr. Taubenberger do? In a fit of desperation, in fear of losing funding, he went back into the possible samples utilized. He and a few other researchers took a single sample out of all 27 samples, only one would work. Based on the presentation of the inflamed lung, he knew that Spanish flu would be in there, of course, because it's not pneumonia, it's not bacteria, so it must be the Spanish flu. Then they created the primer sequences themselves in a process that's not very well defined, just based on previously established H1N1 samples that they're comparing this thing to. Any genetic material that matches it or is similar to it, you're putting into the system to potentially match that primer pair, and then you crank the cycle threshold up to 40, and you had wildly varying annealing temperatures. And just based on this one study, this is the accepted view of the Spanish flu, that it's an H1N1 virus, which this should have been replicated dozens of times in order to confirm these results, by the way. I can't find all those replication studies. Way back in one of my previous lectures, I talked about the problems associated with peer-reviewed research and the lack of replication, and I referenced a study in which they went back and looked at all the landmark sort of cancer papers that were used to dictate, practice guidelines for cancer, and they tried to recreate the results for them. In the overwhelming majority of the time, they couldn't even get close to the results of the original papers. So just because you have one published piece of evidence, and this is a massively flawed published piece of evidence, that doesn't constitute proof of anything. You have to confirm it. Multiple different labs across the world have to confirm the same results before you proceed. And every step of the way with the virus stuff, particularly the stuff that's implemented by people of Jewish ancestry, you don't find those replication studies, because the media that, as I've showed, and at least to a certain extent, has massive Jewish influence over it, promotes these things out into the zeitgeist. People hear these stories, and then it becomes the accepted view, even though it's scientifically bankrupt. And then what follows in the early 2000s? A bunch of swine flu, bird flu, et cetera, et cetera. Why? Because they're relying on the fear that was created during the Spanish flu time period, where millions upon millions died from the Spanish flu. So they retroactively went back and did this study to say that's a variation of H1N1. So at some point in the future, we could have some horrible H1N1 virus, and we need to be prepared for it. And this provides the draconian justification of lockdowns, vaccinations, etc., for fears that one day one of these viruses that's caused by being in close contact with animals and overpopulation and global warming, they've literally said all these things. You can go back and search this if you doubt me. In order to save the planet from a potential future outbreak of H1N1, all of this by Mr. Jeffrey Taubenberger, who's doing presentations in front of the rabbis in 2015.
Which brings us to Paul Offit. He's an American pediatrician specializing in infectious disease, co-inventor of a rotavirus vaccine. Offit is a Maurice R. Hillman Professor of Vaccinology, Professor of Pediatrics at the Perelman School of Medicine at the University of Pennsylvania. He's a member of the Food and Drug Administration's Vaccines and Related Biological Products Advisory Committee, and is a member of the Centers of Disease Control Advisory Committee on Immunization Practices. Offit is a board member of a Every Child by Two and a founding board member of Autism Science Foundation. Offit has published more than 130 papers in medical and scientific journals in the areas of rotavirus-specific immune responses and vaccine safety. He is one of the most public faces of the scientific consensus that vaccines have no association with autism. As a result, he has been the frequent target of hate mail and death threats. You can go back through some of the lectures I referenced, and I do address this and the fact that there has been increased levels of aluminum and things like that detected in the hair of individuals with autism, and increased levels of aluminum on testing in the brains of people that have dementia and other disorders of that ilk. And I've discussed that in some detail once again in previous lectures. It's complicated, the techniques that they use. They often don't appear right out of the blue, and there is more than one cause for autism, in my opinion. There's multiple factors that contribute to this abnormal development that we've talked about. A lot of that has to do with calcium signaling and things like that from other environmental exposures, but suffice it to say, Mr. Offit is not your friend. Son of a shirt maker, he went to his father's sales meetings and reacted negatively to the tall tales told by salespeople, instead preferring the clean and straightforward practice of science. When he was 5 years old, he was sent to a polio ward to recover from clubfoot surgery. This experience caused him to see children as vulnerable and helpless. And motivated him through 25 years of development of the rotavirus vaccine. He decided to become a doctor, the first in his family. Offit earned his bachelor's degree from Tufts University. He completed his residency. One of his mentors was Maurice Hillman, who developed many of the major vaccines in use today. He married Bonnie Fass Offit, who is also a pediatrician. They had 2 children. He's become a leading advocate of childhood immunizations. He was opposed by vaccine critics, many of whom have believed that vaccines cause autism, a belief that has been rejected by major medical journals and professional societies. If you want to get your career ended as fast as possible, associate any sort of link whatsoever to vaccination and autism and see what happens to you. It's not going to work out great. He had colleagues of Fred Clark and Stanley Plotkin, who we'll circle back around to, a pentavalent rotavirus vaccine manufactured by Merck & Co. Since 2006, Rototec has been one of the 2 vaccines currently used against rotavirus. Rototec has been credited by Peter Hotez with saving hundreds of lives a day. Here's an interview that was given. Chop Doc gives inoculation his best shot, February 18, 2015. Q&A with Dr. Paul Offit. This is from the Jewish Exponent, who works to overcome beliefs of anti-vaxxers. And so he says last year a prominent Haredi rabbi, Rabbi Shmuel Kementeski, dean of the Talmudic Yeshiva of Philadelphia, described vaccinations as a hoax. Could you respond to that? Also, how often do you talk with people from the Jewish community who are leery of vaccination? who are leery of vaccinations. First of all, I'm Jewish, and secondly, there's a publication called Peach Magazine. It is a Jewish publication put out by Orthodox Jews making a series of false claims about vaccines causing autism, vaccines causing sudden infant death syndrome, vaccines causing diabetes, attention deficit disorder, multiple sclerosis, all of which have been disproven. Now, I don't recall if in my previous lectures I've addressed the sudden infant death syndrome, but I have addressed some forms of neurodevelopmental disorders. And I have also addressed, with regard to vaccination, some of the work that's been done on the transferring of cancers. And I also addressed that Jonas Salk, in his book Survival of the Wisest, endorses using potentially things like electromagnetic fields and vaccinations as a means of controlling the population. So take from that from what you will, but what are we interested in? The fact that Mr. Offit says I'm Jewish. So we'll come, and he's developing this rotavirus vaccine with Mr. Stanley Plotkin. And then during the period of fears about bioterrorism, he's a member of the CDC's advisory panel to vote against a program to give smallpox vaccines to tens of thousands of Americans. He later argued that the risk of the harm for people getting the vaccine outweighed the risk of getting smallpox in the U.S. at the time. So isn't that interesting? Because if vaccines are 100% safe and effective, why would there be a risk to handing out this smallpox vaccine? After all, the smallpox vaccine back in Edward Jenner's day, which is going to be far less sophisticated, he's literally taking the pus from the pustules on a cow. According to mainstream science, that saved millions of lives, Mr. Jenner's vaccine. But in 2002, a contemporary version of the smallpox vaccine, the risks outweigh the benefit. So they talk about all the numerous rewards he's received, all the publications, etc., etc. But we see that what? He's the main voice in the vaccines don't cause autism debate. He is the developer and promoter of the current vaccination schedule and getting people vaccinated before the age of 2, and the rotavirus vaccine. And he, of course, worked with Peter Hotez and Stanley Plotkin. Who's Peter Hotez? American scientist, pediatrician, and advocate in the fields of global health, vaccinology, and neglected tropical disease control, founding dean of the National School of Tropical Medicine, professor of pediatrics, virology, and microbiology, Baylor College of Medicine. Founding editor-in-chief of PLOS, Neglected Tropical Diseases, co-director of Parasites Without Borders, a focus on those suffering from parasitic diseases in subtropical environments. He was born in Hartford, Connecticut, to a Jewish family. His father, Edward J. Hotez, was a World War II veteran in the United States Navy. Growing up in West Hartford, he graduated from Hall High School. In 1980, he earned a Bachelor of Arts in Molecular Biophysics and Biochemistry, magna cum laude. So he studied parasitology, which is interesting because, permit me a digression here, the drug of choice for treating quote-unquote COVID is a drug called remdesivir, which of course has a high fatality rate associated with it. In studies on Ebola, it was somewhere around 8%, and it caused systemic effects, much like was ascribed to COVID. And the alternative treatment for it was ivermectin, which of course was lambasted in mainstream circles. And my position when describing potential treatments, I described ivermectin. It's a fermentation product of a class of drugs called avermectins that is basically designed to prevent arachnids and certain types of parasites. It's basically poisonous to them. It acts on their central nervous systems, and so it inhibits parasites, which is interesting because it shouldn't have a mechanism of action on a virus whatsoever. But we did see better outcomes with the ivermectin in the studies that we were allowed to do than we saw with the remdesivir. Once again, whenever you look at things in total, there was definitely some doctored-looking studies that were published in that time period. But it's interesting that this guy who's in charge of parasitology, he might know some off-label effects or secondary effects of using this class of drugs and had developed an understanding of it, and he went full board with the vaccination narrative. So it's interesting. With Along with Maria Elena Bottazzi, he led the team that designed the COVID vaccine named Corbevac. Now, having permitted my digression about the ivermectin, what's he working on? He's working on vaccines for hookworm and schistosomiasis and researchers in all of these types of diseases. You don't need vaccines for worms. You just take ivermectin or any other such anti-parasitic which has a known safety profile that's well within acceptable parameters. Now, way back in 2021, I didn't recommend that people take ivermectin on a consistent basis. Taking it for a 6-week time period at a relatively high dose, however, has shown no serious side effects, but I still wouldn't take it chronically, as it's something that has the potential to act on one's central nervous system. And again, whenever we talked about the quote-unquote COVID scenario, the effects were largely mitigated by having appropriate levels of vitamin D in one's body and taking the right type of zinc. And then I correlated that with electromagnetic fields and why some of the symptoms that people were experiencing could be both caused by electromagnetic fields, and some of the potential treatments for quote-unquote COVID could alleviate the symptoms ascribed to electromagnetic fields. But once again, you're going to have to reference those lectures for yourself. But I just find it interesting that he would have the necessary knowledge to know about the treatments for parasites, and he's trying to develop vaccines for them instead.
Vaccines really are like the streaming services of the medical world, because once you get something approved on the schedule, particularly something like a flu shot, you can get that injected. You can have a certain percentage of the population that's going to get that injected in their body every single year. And if there's potentially negative side effects associated with it, well, guess what? The same pharmaceutical companies that are making the vaccinations are making the drugs to treat the side effects that could potentially be caused by the vaccinations. And if you're permanently damaged, well, you have a customer for life. Just saying.
He used his public profile on Twitter to combat misinformation about the COVID-19 pandemic and about vaccines. He appeared as the invited expert on a number of cable news and radio shows, communicating clear messages about the ongoing pandemic. He warned that, contrary to popular beliefs, more young adults than expected would be hospitalized due to the outbreak of COVID-19. We've been trying to appeal to younger adults. He was been the target of online harassment. He tweeted concerns about RFK Jr. sharing misinformation about vaccines. Upon declining the invitation, the Hotez was harassed by their fans with anti-vaccine activist Alex Rosen, who, of course, himself is actually Jewish, and by acting aggressive toward Mr. Hotez, you create a background response to make, quote-unquote, anti-vaccine people look crazy, unstable, and dangerous. He's the parent of an autistic daughter, and he wrote the book Vaccines Did Not Cause Rachel's Autism, My Journey as a Vaccine Scientist. Yes, somebody who works on vaccines and whose daughter is autistic after ostensibly receiving the vaccines surely wants to think about how it could potentially be his fault that vaccines caused that. But I digress. Point being what? The czar of COVID misinformation came from Rockefeller University and stood to make money off of vaccinations, was the go-to media consultant, and of course he's Jewish. Who is their previous contemporary? Well, Mr. Stanley Plotkin, American physician who works as a consultant for vaccine manufacturers such as Sanofi Pasteur, as well as biotechnology firms, nonprofits, and governments. He played a pivotal role in the discovery of a vaccine against rubella virus while working at Wistar Institute in Philadelphia. He is a professor emeritus appointment at Wistar, pediatrics at University of Pennsylvania. His book Vaccines is the standard reference on the subject. So in other words, these individuals are the ones that are determining the reference points by which physicians get their information about vaccines and viruses. He was born and raised in New York City, the son of Jewish parents, Lee and Joseph Plotkin, who immigrated from England. He attended the Bronx High School of Science in New York City, At the age of 15, he read a pair of books that greatly influenced his future education, Aerosmith and Microbe Hunters, once again. Deciding to dedicate his life, Plotkin graduated from Bronx Science in 1948. Plotkin worked on several vaccines: rubella, rabies, rotavirus, and cytomegalovirus. He developed a vaccine for rubella based on RA273 strain of the virus, and also developed by Plotkin using W138 fetal-derived human cell lines, which was released to the public in 1969. What does that mean? That means that Mr. Plotkin was using aborted fetal tissue to develop his vaccine. So the abortion market in the United States for these organs and tissues, etc., is necessary for them to develop new vaccinations to grow out on human cell line. Whenever they talk about religious fanaticism as it relates to vaccinations, there are many other medical products that have a demand for fetal tissue associated with them, but vaccines historically in modern times have had a large demand for these fetal tissues. Now, primarily they're using cell lines that were derived from aborted cells many years ago, i.e. human embryonic kidney tissue from an abortion that took place decades ago, but still it bears worth pointing out the lineage of these medical products and the fact that they're constantly procuring tissues from dead babies to develop medical products. Plotkin worked with Tadeusz Wiktor and Hilary Kaprowski to produce a human vaccine for rabies in the 60s and 70s with a strain gifted to them by Richard Hayflick. Who was Hilary Kaprowski? A Polish virologist and immunologist active in the United States. He authored or co-authored over 875 scientific papers, co-edited several scientific journals. He was born in Warsaw to an educated, assimilated Jewish family. His parents met when Paul Kaprowski was serving in the Imperial Russian Army and moved to Warsaw soon after his marriage in 1912. He attended Warsaw's Mikołaj Rej Secondary School from the age of 12. He took piano lessons. They fled the country after Germany's invasion of Poland. And he was essential in what? The creation of the polio vaccine, based on an orally administered attenuated polio virus. He had focused on live viruses that were attenuated, rendered non-virulent, rather than on killed viruses. This later became the basis for the injected vaccines subsequently developed by Jonas Salk. He developed a polio vaccine by attenuating the virus in brain cells of a cotton rat, a species that is susceptible to polio. He administered the vaccine to himself on January 1948 and in 1950 to 20 children at Letchworth Village, a home for disabled persons in New York. 17 of the 20 children developed antibodies to the polio virus. The other 3 already had antibodies. None of the children developed complications. Within 10 years, the vaccine was being used on 4 continents. Now, again, the documentation for this actual event taking place is quite sparse. So it's largely a verbal account. So think about this. He tested it on himself, then he gave it to 20 kids. That's not a large sample size. And then the media apparatus riles everybody up enough that this winds up getting distributed to multiple continents around the world. Do you think if it's just these guys, organic say-so or know-how about this, that you could get this distribution network? No, this has to be promoted. He also worked with Albert Sabin, Polish-American medical researcher known for developing the oral polio vaccine. Abraham Stapergin was born in Bialystok, Russian Empire, to Polish-Jewish parents, Jacob Stapergin and Tilly Krugman. In 1921, he immigrated with his family on the SS Lapland, which sailed from Antwerp, Belgium, to the port of New York. In 1930, he became a naturalized citizen of the United States, changed his name to Sabin, as well as assuming the middle name Bruce. He was interested in virology and changed majors. He received a bachelor's degree in science in 1928. Went to Bellevue Hospital, conducted research at the Lister Institute, and then joined the Rockefeller Institute for Medical Research. Hopefully people have been picking up on the Rockefeller connection this entire time, because all of these ideas, as far as it relates to modern virology and its development in the early 1900s to late 1800s, have flourished under Rockefeller Institution leadership. So he lived in Israel, serving as a president of the Weizmann Institute of Science at Rehovot. He worked in the United States as a research professor at the Medical University of South Carolina. With the menace of polio growing, and other researchers, Jonas Salk and Hilary Kaprowski and H.R. Cox in New York, sought a vaccine to prevent or mitigate the illness. Salk developed an inactivated polio vaccine, which was released for use in 1955. It did not prevent initial intestinal infection. By carrying out autopsies on polio victims, Sabin was able to demonstrate poliovirus multiplied and attacked the intestines before it moved to the central nervous system. Now, in the lectures that I showed you, in the Jonas Salk, I showed you a clip in which they were literally going around spraying DDT over everything to supposedly prevent the polio virus. And of course, one of the secondary effects of DDT exposure is the potential for flaccid paralysis. So he develops this oral vaccine, tested on 100 million people in the USSR, parts of Eastern Europe, Singapore, and Mexico, and the Netherlands.
So we saw that Hotez and Offit are these vaccine advocates for the schedule, and they deny autism. And they were both working with Stanley Plotkin, who was working with those other figures that contributed to the development of the polio vaccine. Now, way back in my 5th lecture on vaccination, I showed you this clip of Mr. Stanley Plotkin under oath discussing the usage of experimental vaccines and fetal tissue on their development.
So this study involved 74 fetuses, correct?
I don't remember exactly how many.
Turn to page 12 of the study.
Yeah, 76.
76.
Mm-hmm.
And these fetuses were all 3 months or older when aborted, correct?
Yes.
Okay. And these were all normally developed fetuses, correct?
Yes.
Okay. These included fetuses that were aborted for social and psychiatric reasons, correct?
Correct.
What organs did you harvest from these fetuses?
Well, I didn't personally harvest any, but a whole range of tissues were harvested by co-workers.
Okay. And these pieces were then cut up into little pieces, right?
Yes.
And they were cultured?
Yes.
Okay. Some of the pieces of the fetuses were pituitary gland that were chopped up into pieces to okay, included the lung of the fetuses?
Yes.
Okay, included the skin?
Yes.
Kidney?
Yes.
Spleen?
Yes.
Heart?
Yes.
Okay. And tongue?
I don't recall, but yeah, probably yes.
So I just want to make sure I understand. In your entire career, and this was just one study, so I'm going to ask you again. In your entire career, how many fetuses have you worked with?
Well, I don't remember the exact number, but quite a few when we were studying them originally, before we decided to use them to make vaccines.
Do you have any sense? I mean, this one study had 76. How many other studies did you have that you used aborted fetuses for?
Oh, I don't remember how many.
You're aware? Are you aware that one of the objections to vaccination by the plaintiff in this case is the inclusion of aborted fetal tissue in the development of vaccines, and the fact that it's actually part of the ingredients of vaccines?
Yeah, I'm aware of those objections. The Catholic Church has actually issued a document on that, which says that individuals who need the vaccine should receive the vaccines regardless of the fact, and that I think it implies that I am the individual who will go to hell because of the use of aborted tissues, which I am glad to do.
Okay. Do you know if the mother is Catholic?
I have no idea.
Okay. So
But she should consult her priest.
if she has if she's in fact Christian, I guess, right? In any event, so we have 76 in this study. Would you approximate it's been a few hundred fetuses?
Oh, no, I don't think it was that many.
Okay.
It was probably not many more than in this paper. And I should stipulate that we had nothing to do with the cause of the abortion.
Some of these were for psychiatric institutions, correct?
Actually, all I can say is that the fetuses that I personally worked with actually came from Sweden, from a Swedish co-worker. And so I, in no case, was able to determine what exactly the reason for the abortion was.
I'm just asking you, some of the fetuses that you did use did come from abortions from people who were in psychiatric institutions, correct?
I don't know that. What I'm telling you is that I got them from a coworker, and if it's stated in the paper, it's true, but otherwise I do not know.
Like, so if it's in the paper, you don't contest it, right?
I don't contest it. No.
Okay. Have you ever used orphans to study an experimental vaccine?
Yes.
Have you ever used the mentally handicapped to study an experimental vaccine?
I don't recollect ever doing studies in mentally handicapped individuals. At the time, in the 1960s, it was not an uncommon practice.
So, you're saying, I'm not clear on your answer, I'm sorry. Have you ever used the mentally handicapped to study an experimental vaccine?
What I'm saying is I don't recall specifically having done that, but that in the 1960s it was not unusual to do that, and I wouldn't deny that I may have done so.
Okay. There's an article entitled "Attenuation of RA27.3 Rubella Virus in WI38 Human Diploid Cells." Are you familiar with that article?
Yes.
In that article, one of the things it says is 13 seronegative, mentally retarded children were given RA27.3 vaccine.
Okay, well, then that's, in that case, that's what I did.
Okay. Have you ever expressed that it's better to perform experiments on those less likely to be able to contribute to society, such as children with handicap, than with children without or adults without handicaps?
I don't remember specifically, but it's possible. And again, I repeat that in the 1960s, that was more or less common practice. I've since changed my mind, but that was a long time ago.
Do you remember ever writing to the editor of Ethics on Human Experimentation?
I don't remember specifically, but I may well have.
Okay. I'll mark this. Yeah. was marked as Exhibit 43. Do you recognize this letter you wrote to the editor?
Yes.
Okay. Did you write this letter?
Yes.
Okay. Is one of the things you wrote, the question is whether we are to have experiments performed on fully functioning adults and on children who are potentially contributors to society, or to perform initial studies in children and adults who are human in form but not in social potential.
Yes.
Okay. It may be objected that this question implies a Nazi philosophy, but I do not think that it is difficult to distinguish non-functioning persons from members of ethnic, racial, economic, or other groups.
Mm-hmm.
Okay. Have you ever used babies of mothers in prison to study an experimental vaccine?
Yes.
Have you ever used individuals under colonial rule to study an experimental vaccine?
Yes.
Did you do so in the Belgian Congo?
Yes.
Did that experiment involve almost 1 million people?
Well, all right. Yes. Okay.
Did you ever visit what was the Belgian Congo and Urundi?
Yes.
How many times?
Once.
This brings us to the COVID vaccines themselves. This is from the Charles Lozier Institute, update COVID-19 vaccine candidates in abortion-derived cell lines. So they give you this flow chart.
The Jews were behind the invention and creation of these COVID vaccines that are going to save humanity now from what would be an unbelievable, unspeakable catastrophe. And you'd have a catastrophe here where untold numbers, untold millions would die, and the economies of the entire world would collapse, would crash and collapse if this thing went on for another few years, which is what would happen if not for Jewish brains. It was Jewish brains that created these two COVID vaccines. And, by the way, just to show you who specifically we're speaking about, so we're not making this up, the Moderna vaccine. Okay, the Moderna vaccine, the chief medical officer in charge of research and development, the person who was in charge of putting it all together is the Israeli scientist Tal Zaks, who is an Israeli who does interviews on Israeli media in Hebrew. He left Israel. He's one of these Israelis who left Israel because he's greedy and he wants more money. And he claims he wants to go where the science permits him to do the most good. So full of crap. Pfizer. Who's the CEO of Pfizer? In other words, the person who heads the company. Albert Bourla, who is a Greek Jew. Albert Bourla is a Jew from Greece. And the chief Pfizer scientist who worked on the research, in other words, the company, the person who got the financing, everything else, was Albert Bourla, the CEO. He's the one who was the executive in charge, who organized everything to enable financing and enable all the work and research to go forward the way it's supposed to go forward. That took Jewish brains. And then, who was the chief scientist? Chief scientist is Mikael Dolsten, a Jew from Sweden. The Jews were in charge of the research, the science. They were the scientists. They're the main researchers. The Jews are the ones that led the company efforts and the ones that brought these vaccines, that created these vaccines. Both COVID vaccines that are now going to rescue humanity, both were created by Jews.
That little clip has a different connotation now, doesn't it? So I'm also going to show you here some of my lectures that I covered in the past talking about the COVID vaccines really quickly.
The media coverage of the COVID vaccines themselves, and we saw that a private company that's basically associated with the military-industrial complex was the one that was setting up these contracts in a complex manner and resulted in them being protected in such a format that you're not able to initiate a Freedom of Information Act request to identify where these contracts are, how these contracts were set up. In these two lectures, I discussed in detail the long history of the failures of mRNA vaccines and specifically with the company Moderna. So in 2005, we supposedly had identified this effective technology for modifying and preventing inflammatory reactions and blah, 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 blah. But as I already showed you, as recently as 2017, we had abject failures, and they had reverted to using this technology for the Zika virus stuff, which ultimately, I would argue, still wasn't successful, even after over a decade. So why would we think that in 2020 this is going to be so successful? And for those that doubt any of the claims that I make about the COVID vaccine itself, here is where I broke down this efficacy and where they got this 95% effective number. Now, obviously, you would have to be a complete and utter moron to still believe in 90% effectiveness. Maybe you're on your 20th booster at this point. I don't know. But at this point, and in these lectures specifically, is where I discuss how all of these physicians and doctors that were recommending you this shot were either shills or so brainwashed that they weren't even attempting to interpret the data. So, and I used some very specific examples, including some that people seem to be on board with now. Mr. John Campbell. Everybody seems to like Mr. John Campbell on YouTube, Dr. John Campbell. And you can go back and watch this clip in which I show him talking about how great the vaccine was. Anybody that knows how to interpret research would know that this vaccine testing was an abject failure and had provided no evidence of transmission. In the early trials between both sets of data for both Pfizer and Moderna, they only captured one COVID death. That means you have to hand out tens of thousands of shots to prevent one COVID death. There's no way you can ascribe, minus all the people that were lost follow-up between the numerous trials and the fact that they vaccinated the control group shortly after the study. But this is where you would find that information. And here's where we start the discussion on vaccine injuries and the various vectors for that, including various compounds and their known negative effects. Now, it's at this point that I'll say, we don't know necessarily exactly what's in this shot, and I don't think that it's the same product that was given to everyone. And just because there's patents for something doesn't necessarily mean they're used. After all, this is being treated more like a trade secret than a patent. So my argument would be that we don't know exactly what everybody received with these shots. So 100% anticipation of the effects is not going to be likely, particularly with the fact that they were kept on ice for a significant period of time. So, and there's multiple technologies that could benefit from that preservation period. So again, we're dealing with numerous factors here.
So here we have this article, how Israel became a world leader in vaccinating against COVID-19. Badly hit by the coronavirus, Israel demonstrated the first two vaccines to more than 10% of its population. This is by June 1st, 2021, and Benjamin Netanyahu led the charge. Less than 1% of the United States and only a small fraction of the population in many European countries received a vaccine dose by the end of 2020. So it wasn't they were, quote-unquote, ahead of schedule. Health Minister Yuli Edelstein said in an interview that Israel had entered into negotiations with drug makers as an early bird and that companies were interested in supplying Israel because of its HMO's reputation for efficiency and gathering reliable data. So they had a separate pipeline than the rest of the world. Michael Dolston, the Jewish immigrant leading Pfizer's vaccine charge, hopes the U.S. stays a melting pot. Head scientist at Pfizer heard the news last week from the COVID-19 vaccine. He had been helping to develop the better part of a year, was over 90% effective. He and his colleagues literally leapt for joy at a corporate office in Connecticut. This may turn out to be one of the biggest medical advances of the past 100 years, Dolston said by Zoom. It is a proud moment for Jewish immigrants like himself who have contributed to American scientific innovation. A lot of the great breakthroughs in America have come from people that immigrated, he said, pointing to Albert Einstein. There has been a strong Jewish tradition around contributing to humanity and a strong tradition within medicine. Albert Bourla is a Jew from Thessaloniki, Greece, the chief medical officer for Moderna and competing drug maker that announced Monday that its COVID-19 vaccine was nearly 95% effective, is an Israeli immigrant named Tal Zaks. In the person in charge of Pfizer's scientific development team was Jewish, and the individual in charge of Moderna's developmental team was Jewish, and Pfizer's CEO is, of course, Jewish. COVID-19 vaccines have Jewish links. So again, here's Albert Bourla. I'll show you a quick clip of him whenever we get done from this.
I want to ask you also about a very important issue of vaccine hesitancy. Even folks so quickly, what do you tell those folks who might be saying, I'm going to wait a few months before I get this one?
I would tell them I wish that the situation was not so critical so that they can have the luxury to think about it. But the situation is as deadly as it could be right now. The situation is as deadly as it could be.
You have not yet had your shot. When do you plan to get it? You have not yet had your shot. When do you plan to get it?
This is a vaccine that was developed without cutting corners from a company with 171 years of credentials. The only sensitivity here, Meg, is that I don't want to have an example that I'm cutting the line.
What do you tell those folks who might be saying, I'm going to wait a few months before I get this one?
I would tell them I wish that the situation was not so critical so that they can have the luxury to think about it. I am 59 years old, in good health. I'm not working in the front line. So my type is not recommended to get vaccination now. So the situation is as deadly as it could be right now.
Descended from Sephardic Jews who fled from Spain in 1492. They had achieved 90% effectiveness. Again, they mention Tal Zaks here as well. Evidence of neutralizing antibodies in quantities comparable to those who had survived COVID-19. So again, I just want to highlight the ridiculousness of this where they're saying, look, we're producing quote-unquote antibodies at a level that's similar to people that have quote-unquote already had the virus. Again, you can go back and reference my previous lectures that I showed you on that. And then here's a picture of Tal Zaks, who's Moderna's chief medical officer. Pfizer negotiated a production delivery deal with the U.S. government worth up to $1.95 billion to secure an initial 100 million doses of its vaccine, with an option to purchase an additional 500 million. Pfizer's vaccine needs to be kept very cold at temperatures colder than minus 100 degrees Fahrenheit when transported, while Moderna's reportedly can be transported and stored at less extreme sub-zero temperatures. So, of course, the question is why? And then once again, they touch on the aggressiveness of the Netanyahu approach and talk about some accusations that they say are anti-Semitic. Who are the Jews behind the coronavirus vaccines? This is November 2020. One can't escape their presence in the forefront of leading companies in a race to find a vaccine that will prevent the coronavirus. So again, they reference this Ben-Gurion University and Tal Zaks and Albert Bourla. There have been vile anti-Semitic accusations against Bourla and Jews in general, similar to negative comments made by U.S. President Donald Trump about the Chinese, which is interesting when you look at this because, again, Trump's son-in-law is Jewish. Bourla comes from a family of Sephardi jewelers. In April 2019, Bourla was named the most preeminent Greek leader of the global pharmaceutical industry. And I just want to remind people of this is Jerusalem Post with their COVID death counter. Look at how bad it was. Global deaths in the U.S., 379,365 deaths. And I don't know how well you've kept up on the all-cause mortality above and beyond what's been expected, but 2021 was worse than 2020. 2022 was worse than 2021, and 2023 was worse than 2022. Again, we locked down the entire world because of this, and we gave them remdesivir and put them on ventilators and all those things that I've again covered in previous lectures 2 years ago. But now that more people are dying than there was during the height of the pandemic, nobody cares. So with all of that said, we're going to continue with part 3 with our discussion of Jewish and Israeli involvement in the development of the COVID vaccine.